Hi there, thank you for joining me in this talk. In animal kingdom, competition for limited resources is a common ecological interaction. Two insect parasitoids whose eggs and larvae develop in or on the body of other arthropods, whose resources are not immediately consumed and remain in the habitat. It can be subsequently parasitized by a female of the same species, an act called superparasitism, or by a different female species. It is called multiparasitism. Some parasitoid females can recognize parasitized hosts, especially when the host was parasitized by a female of the same species or a closely related species. The decision to accept or reject a parasitized host depends on a combination of ecological and physiological parameters and the fitness consequences. Oviposition in an already parasitized host by con or heterospecific could be advantageous when there is no time to search unparasitized hosts, when unparasitized hosts are not available, or when female can kill eggs or larvae present on the host. When multiparasitism occurs, the relationship is much more asymmetric than superparasitism, and some species may always win when in competition with an intrinsically weaker species, especially to solitary parasitoids. In this illustration, a caterpillar is parasitized by two different types of parasitoids, first by a solitary koinobiont endoparasitoid on the left, and then by a solitary idiobiont ectoparasitoid on the right. Koinobiont endoparasitoids are those whose adult females deposit her eggs inside the host, and the larva develop inside the host, which continues to feed, grow, and defend itself during the course of parasitism. Idiobiont ectoparasitoids are those whose adult females deposit her eggs outside the host, which is killed or paralyzed permanently at the time of oviposition, and larvae feed externally. Idiobiont ectoparasitoids are often physiological competitors superior to koinobiont endoparasitoids because they attack later stages, produce larger eggs, and inject venom that paralyze endoparasitoid larvae as well as the host itself, and larvae feed externally and do not have to deal with internal metabolic host defense. Here we studied the interaction between two forest parasitoid species that attack the same host species. Ibalia leucospoides and Megarhyssa nortoni are two of the most commonly used biological control agents against population of Cyrex noctilio, an economically important woodwasp pest of pine plantations on southern hemisphere. Ibalia leucospoides is a koinobiont endoparasitoid that attacks eggs and early instars of hosts during summer and early autumn. Megarhyssa nortoni is an idiobiont ectoparasitoid that attacks late instars and pupa of hosts during spring and early summer. Here we hypothesized that Megarhyssa nortoni is able to multiparasitize host patches attacked by Ibalia leucospoides because it is a physiological superior competitor. The resulting prediction is that oviposition and host-finding behavior of Megarhyssa nortoni females are similar both in parasitized and unparasitized hosts by Ibalia leucospoides. The aim of this study was to investigate the foraging behavior of Megarhyssa nortoni females in relation to parasitized host patches by Ibalia leucospoides. The experiment consisted in releasing a female of Megarhyssa nortoni in an experimental arena and analyzing the patch time allocation and oviposition behavior according to one of two treatments: 1. Unparasitized host patch and 2. Parasitized host patch by Ibalia leucospoides. Each host patch was a vine log attacked by 4 host females of Cyrex noctilio during 48 hours. Then part of these patches was exposed separately to 4 Ibalia leucospoides females during 48 hours to create parasitized host patch treatments. We analyzed different behaviors displayed by Megarhyssa nortoni females in order to know their behavioral response to treatments. The total number of exploratory behaviors as walking and antennal contact, the oviposition behavior, and resting and grooming behaviors were similar at parasitized and unparasitized host patches. In many parasitoid species, the reproductive success or the number of offspring laid is limited by time. Therefore, time is an important variable to parasitoids. We analyzed the total time spent on patches by Megarhyssa nortoni females, and we did not find significantly differences between unparasitized and parasitized host patches. Some parasitoids prove a patch with the ovipositor, allowing them to accept or to reject a host. Therefore, we analyzed the duration of each oviposition, and we again did not find any difference between treatments. As a discussion, we could say that our results were consistent with the hypothesis presented, suggesting that Megarhyssa nortoni is able to multiparasitize host patches attacked by Ibalia leucospoides. Results showed that behavioral responses of Megarhyssa nortoni females to unparasitized and parasitized host patches by Ibalia leucospoides were similar in both situations. The first parasitoid attacking a host could monopolize resources with a consequence on host nutritional quality required to the second parasitoid. Therefore, it would be interesting to analyze the fitness cost of multiparasitism to Megarhyssa nortoni. Multiparasitism does not necessarily imply that coexistence within shared habitat is impossible. But from a biological control perspective, it always has a negative impact except when the species of interest is intrinsically superior in competition. The potential ability of Megarhyssa nortoni to multiparasitize could explain the success of establishment in different regions where it was introduced to control the exotic wood wasp pest. Thank you.